you have any doubts, please type in the chat box. Okay, and we will discuss it. Okay, what we have seen in last class. This is the overview of digestive system that we are going to deal with. Organs of digestive system and the digestive activity in all these organs, and the role of digestive enzymes, and the role of various hormones of digestive system and phases of digestion. Okay, and we have seen the functions of digestive system: ingestion, secretion, motility, digestion, absorption, and defecation. And we have seen how ingestion takes place and about all these things. And we have seen the digestive system consists of this mouth, pharynx, esophagus, stomach, small intestine, and large intestine, and the accessory digestive organs. And we are dealing with one by one. We have seen this. What are the layers of the gastrointestinal tract? What's the nerve supply? The organs of the digestive system. We have seen the mouth, salivary glands, the composition of saliva, digestive activities that occur in the mouth, um, in the cheeks and lips, salivary glands, what is the tongue does, what do the teeth does, what is the mechanical and chemical digestion that occurs in the mouth. And we have seen about the pharynx, esophagus, the deglutition, and we have seen about the digestive activities in the pharynx and esophagus, and we have seen the role of salivary amylase and lingual lipase. And we have seen the stomach, the layers of the stomach. Here you can see this is the fundus for your information. This is the cardia, and you can see the body, and you can this is the pylorus. Just remember these places, okay? And you can see the duodenum here. And we have seen the composition of gastric juice. What is the functions of the stomach? We have seen the activities of the parietal cells, all these things, G cells, chief cells, all this. Okay, guys. Hope you got a quick revision of everything what we have seen in the previous class. Okay. And now what we will do, I'll quickly show you these things. You can see here mucous neck cells, parietal cells, and chief cells. These are the exocrine cells. And you can see the G cells. These are the endocrine cells. You will see about the enterogastrin in further slide. Just remember that it is a combination of hormones released by the intestine that inhibits the gastric secretion. And you know, this mucous cells and mucous neck cells, what they'll do is they'll secrete mucus. And what is the function of this mucus? It forms the protective barrier that prevents digestion of stomach wall. And also it functions it also performs the function of absorption. Like it absorbs water, ions, short chain fatty acids, and some drugs that enter the bloodstream. Okay. And parietal cells, you can see here clearly. What is the function of these parietal cells? What does this parietal cells do? They secrete the intrinsic factor, and also they secrete the hydrochloric acid. What is the function of this intrinsic factor? I've told you in last class, it is needed for absorption of vitamin B12. And vitamin B12 is used in red blood cell formation or erythropoiesis. Okay. And what does this hydrochloric acid do? This hydrochloric acid will kill the microbes that are present in the food, and it denatures the proteins and converts the pepsinogen into pepsin. Okay, next we'll be seeing about the chief cells. These chief cells, no, they secrete pepsinogen, and is also they secrete the gastric lipase. Okay, actually pepsinogen is the inactive form. Pepsin is the activated form which breaks down proteins into the peptides. Another is gastric lipase. Okay. And what does it do? It splits the triglycerides into fatty acids and monoglycerides. And this gastric lipase, what does it do? This is the function of gastric lipase. And this is pepsinogen, which is the inactive form converted into pepsin, and it performs this function. Okay. Next, we'll see this G cells. These G cells secrete a hormone called as gastrin. It stimulates the parietal cells to secrete the hydrochloric acid and chief cells to secrete the pepsinogen. Okay. Hope you're clear. You can see the gastric glands, this is lamina propria, this muscular mucosa. This muscular is, you know, right? It is helpful for the mixing. Okay, the gastric peristaltic movements. Okay. And this pyloric sphincter opens into the passage of, it will permit the passage of chyme into the duodenum. Okay. Now, quickly we will see the phases of secretion of gastric juice. That is very important for us to understand. Last class we have seen what is the composition of gastric juice. It contains water, mineral salts, mucus that is secreted by the goblet cells in the glands, and on the stomach surface it contains hydrochloric acid, intrinsic factor, and also inactive enzyme precursors like pepsinogens, which are secreted by the chief cells of the chief cells in the glands. Okay, now we will discuss about the secretion of gastric juice. You remember, gastric juice is the combination of the secretions of the mucus cells, parietal cells, and chief cells. And there is always a small quantity of gastric juice present in our stomach, even when there is no food. Okay, this is known as fasting juice. And secretion reaches its maximum level about 1 hour after you take a meal, and then slowly it will start decreasing after about 4 hours. Okay. And to tell you, the secretion of gastric juice in three phases. That is cephalic phase. You can see it here. The cephalic phase, the gastric phase, and the intestinal phase. Now we will deal with the cephalic phase. This flow of juice occurs before food reaches the stomach. And why it happens? And it is due to reflex stimulation of the vagus nerve initiated when there is sight of smell, taste, thought, and the taste of food. And suppose if there is no vagus stimulation, if vagus nerves have been cut, so this phase of gastric secretion will not be there in that case. Cephalic phase is not there. Now gastric phase. What will happen in gastric phase? You can see it here. In gastric phase, what is happening is when stimulated by the presence of food, the enteroendocrine cells in the pyloric antrum, I've shown you where is antrum, and duodenum secrete gastrin. Okay. A hormone which passes directly into the circulating blood. Gastrin passes directly into the bloodstream. This gastrin is circulating the blood, which supplies the stomach, stimulates the gastric glands in the stomach to provide more gastric juice. In this way, what it will happen is the secretion of digestive juice is continued even after the completion of the meal and at the end of the cephalic phase. And gastrin secretion will be suppressed when the pH in the antrum falls to about 1.5. That is acidic. It will not go lower than that. So there will be no secretion of gastrin and there will be no stimulation of HCl and all. Next one is the intestinal phase. What will happen in this intestinal phase? When the partially digested contents of the stomach reach the small intestine, a hormone called enterogastrone is produced by this endocrine cells in the intestinal mucosa. It slows down the secretion of gastric juice and reduces the gastric motility. And the two of the hormones which form this complex are secretin and cholecystokinin. Okay, when the food is passing through intestine, the gastric motility should decrease because there will be no need of gastric functions when food passes from stomach to the intestine. Okay, so by slowing the emptying rate of the stomach, the contents of the duodenum become more thoroughly mixed with the bile and pancreatic juice. Okay, this phase of gastric secretion is mostly when the meal has a high fat content. The rate at which the stomach empties depends to a large extent on type of food you eat. If you have a carbohydrate meal, what will happen? It leaves the stomach in 2 to 3 hours. A protein meal remains longer and a fatty meal remains in the stomach the longest. That is the reason when you eat a fat meal, you don't feel hungry for a long time. So compared to carbohydrate, proteins and fats, if you're eating carbohydrates, you feel hunger fast. You'll be like eating more and more. That's why when people who are doing dieting and all, they go for a high fat meals because long time it will be in stomach and for most of the time your stomach is full and you will not feel hungry, and compared to protein, and then leaves the carbohydrates. Okay.
Someone wants to repeat it again, Dr. Pravallika.
What?
Someone wants to repeat it again.
The last part?
Yes.
Okay. See, the rate at which I told you the stomach will empty the.
Dr. Pravallika, you have to open your mic.
Okay. Can you hear, guys, now?
Yeah. And now everyone can even interact. If they have questions, they can talk to Dr. Pravallika as well.
You can type in the chat box once the topic is over. While going to next topic, we'll discuss, so that there will be no disturbance, because 156 students are there.
Sure, done.
Let it not be a confusion.
Yeah.
And if more questions are increasing, you can stop me and ask me. Okay, guys, carefully listen. I told you, when people are going for diet, okay, so many people go for weight loss and all this. At that time, you can see, you can tell that keto meal, keto diet will keep you full for longer, lighter, and also, for most of the time, you don't have cravings. Your stomach is full. But when you eat a carbohydrate meal, after some time, you can eat, you can dump something in your stomach. This is why, because when a person eats a carbohydrate meal, it leaves the stomach very fast. And a protein meal remains longer, and fatty meals remains the longest in the stomach. So gastric emptying time is more. It will delay in case of fatty meal when compared to protein and carbohydrate meal. You will understand all this by the time we complete the presentation, when we go step by step. Okay. Somebody's mic is on, Dr. Ajit.
Yes, now everyone is on mute, so you can open your mic and carry on, Dr. Pravallika. Hello. Yes, Dr. Pravallika, now you can open your mic and can continue. Nobody will speak now.
Yeah, now is it clear? I think no sounds, right? Please mute your mics, guys. Now we will be dealing with the mechanical and chemical digestion in the stomach. Several minutes after the food enters the stomach, okay, waves of peristalsis pass over the stomach every 15 to 25 seconds. And few peristaltic movements are observed in the fundus, which primarily has a storage function. But most of the waves, okay, they begin at the stomach and intensify as they reach the antrum. Okay, at the fundus, you will have more peristaltic movements, which will start there, and then they start increasing as the food reaches the antrum. Okay, and each peristaltic wave moves this gastric contents from the body of the stomach down into the antrum, and this process is known as propulsion. Okay, and the pyloric sphincter, what it does, normally it remains almost but not completely closed, because food particles in the stomach initially are too large to fit through the narrow pyloric sphincter. I have told you last time. They're forced back into the body of the stomach, and that is known as retropulsion. Okay. And the net results of these movements, okay, is that gastric contents are nicely mixed with the gastric juice, and eventually they form to some soupy liquid type of thing called as chyme that can be easily moved through this pyloric sphincter, because the size of the particles will be reduced. Once the food particles in the chyme are small enough, okay, that they can pass through the pyloric sphincter, it will move, and this process is this phenomenon of moving of this chyme is called as gastric emptying. It is a very slow process. Only about 3 ml of chyme moves from the pyloric sphincter each time. And foods may remain in the fundus part of the stomach for about 1 hour, without even being mixed with the gastric juice. During this time, what will happen? This digestion by the salivary amylase from the salivary gland will continue. when this acid will start mixing with this food, what will happen at the time? Slowly, salivary amylase will get inactivated, and lingual lipase that is produced by the tongue, which will start to digest the triglycerides into fatty acids and diglycerides. Okay, that is what happens. And another thing is, what will happen? This HCl secretion will be happened by the parietal cells that can be stimulated by several sources. Okay, I'll show it here. 1 second. You can see here. This HCl secretion by the parietal cells can be stimulated by several sources. That means acetylcholine that is released by parasympathetic neurons, gastrin secreted by G cells, and histamine, which is a paracrine substance released by mast cells in the lamina propria. Okay, acetylcholine and gastrin stimulate the parietal cells to secrete more HCl in the presence of histamine. What it means? Histamine gets synergistically enhancing the effects of acetylcholine and gastrin. And all these receptors are present in the plasma membrane of the parietal cells. Okay. And guys, the strongly acidic food of the stomach, okay, it kills many microbes in the food. And HCl partially denatures proteins in the food and stimulates the secretion of hormones that promote the flow of bile and pancreatic juice. The only proteolytic, that is the protein digesting enzyme that is present in the stomach, is pepsin. And pepsin is most effective in very acidic environment of the stomach, that is pH 2. And it becomes inactive at a higher pH. I'll ask you, what keeps pepsin from digesting the protein in the stomach cells along with the food? Okay. Firstly, I told you pepsin is secreted in the inactive form called as pepsinogen, and in this form it cannot digest the proteins in the chief cells. So what it will happen? Pepsinogen is not converted into active pepsin until it becomes in contact with the hydrochloric acid that is secreted by the parietal cells or active pepsin molecules. And secondly, the stomach epithelial cells are protected from gastric juice by layer which is 1 to 3 millimeter thick of alkaline mucus that is secreted by the mucus cells and mucus neck cells that I have told you. There's another enzyme in the stomach, that is gastric lipase, which splits the triglycerides, okay? That is that all the fats and oil content that you eat into fatty acids and monoglycerides. I'll show it to you here. Now, when we go to small intestine, what will happen is the small intestine is continuous with the stomach at the pyloric sphincter and leads to a large intestine at the ileocecal valve. Okay. It is a little over 5 meters long and lies in the abdominal cavity surrounded by the large intestine. What are the small intestine compromises of? We should know this one. Small intestine contains duodenum, jejunum, and ileum. What is this duodenum? It is a, I'll show you in the picture. This duodenum is nothing but a 25 centimeter long, okay, and it is around, it will be surrounding around the head of the pancreas. And the secretions from the gallbladder and pancreas are released into the duodenum through hepatopancreatic ampulla, and the opening into the duodenum is guarded by the hepatopancreatic sphincter that is nothing but the sphincter of Oddi. That is not seen here, guys. Next one is the jejunum. Jejunum is the middle section of the small intestine, and ileum is the terminal section. Okay. And the small intestine contains the peritoneum, mucosa, and mucosa has villi. Villi, the permanent circular folds, like unlike the rugae of the stomach, are not smoothed out when small intestine is distended. They will promote the mixing of chyme as it passes along it. You can see here. Now we'll talk about the blood supply. Superior mesenteric artery will supply blood to the intestine. All small intestine is supplied with blood supply through superior mesenteric artery, and the venous drainage is from superior mesenteric vein. Okay, and the nerve supply is by both sympathetic and parasympathetic nervous system. It will be there. And now we will talk about the intestinal juice. About 1500 ml of intestinal juice are secreted daily by the glands of the small intestine. What does it contain? Water, mucus, mineral salts, like enzyme enterokinase, that is enteropeptidases, and the pH is between 7.8 to 8, that is alkaline pH. And we'll see the what are the functions of the small intestine. You know, as the chyme moves, that's the onward movement of the contents is produced by the peristalsis. Peristalsis, because of peristalsis, what will happen? There will be the movement of the chyme or the contents of the contents present in the gastrointestinal tract, and there will be secretion of intestinal juice. And it will complete the chemical digestion of carbohydrates, proteins, and fats is completed in the enterocytes of the villi, and there will be protection against the infection by the microbes that have survived the antimicrobial action of the hydrochloric acid in the stomach. Usually hydrochloric acid will kill the microbes, but there will be some microbes, the acid labile ones, which are not killed in the stomach, that will be killed here. Okay, so there will be a protection against the infection. How it will happen? There will be some lymphatic follicles which are aggregated in the intestine, that will provide the immunity by protecting against the microbial infections. And there will be secretion of cholecystokinin and secretin, and absorption of nutrients takes place in the small intestine. And now let us see about the chemical digestion that happens in the small intestine. When acid chyme passes into the small intestine, it is mixed with the pancreatic juice, bile juice, and intestinal juice, and is in contact with the enterocytes of the villi. And I told you, in the small intestine, the digestion of all the nutrients is completed. See, you can see the villus here. And these are the microvilli. This is the enterocyte. Okay. And this is the goblet cells. And this is the goblet cells. And next one, before we deal with it, what happens in the small intestine about the digestion of carbohydrates, digestion of fats, and all these things, and digestion of proteins. Let us see, we have told that it is mixed with the pancreatic juice, bile juice, and intestinal juice. And we will see what is this pancreas first. Okay, the pancreas is a pale gray gland weighing about 60 grams. It is about 12 to 15 centimeter long and is situated in the epigastric and left hypochondric regions of the abdominal cavity. The pancreas is both exocrine and endocrine. So what is the exocrine function of the pancreas? And this exocrine function, we'll talk about this exocrine and endocrine function of pancreas. What is this exocrine pancreas? It consists of large number of lobules that are made up of small alveoli, the walls of which consist of secretory cells. Okay, secretory cells. And the function of pancreas is to produce pancreatic juice containing enzymes that digest carbohydrates, proteins, and fats. And this is the exocrine function of the pancreas. And what is this endocrine function of the pancreas? And you know, throughout the pancreas gland, there are groups of specialized cells called islets of Langerhans, that is pancreatic islets. They have no ducts, so the hormones will diffuse directly into the blood. Okay, and they help in secretion of the hormones like insulin, glucagon, which are principally concerned with the control of blood sugar. Next one, you can see the compositions of the pancreatic juice. Pancreatic juice enters the duodenum at the hepatopancreatic ampulla, and it consists of water, mineral salts, enzymes like amylase, lipase, and inactive enzyme precursors like trypsinogen, chymotrypsinogen, and procarboxypeptidase. And what is the function? I told you it helps in digestion of proteins, digestion of carbohydrates, digestion of fats, the small intestine functions. So what will happen here in small intestine? I have told you that it has all these functions. So digestion of proteins. We'll discuss about the digestion of proteins. These proteins are broken down into amino acids. I told you that trypsinogen and chymotrypsinogen are the inactive enzyme precursors which are activated by the enterokinase, that is enteropeptidases, that is an enzyme in the microvilli. It will convert into proteolytic enzymes trypsin and chymotrypsin. And this trypsin and chymotrypsin, what they will do, these enzymes will convert the polypeptides to tripeptides, dipeptides, and amino acids. Likewise, in the intestine, digestion of protein happens by converting proteins into the amino acids. Okay. Next one will deal about the digestion of carbohydrates. And pancreatic amylase, it converts all the digestible polysaccharides, that is nothing but starches, which are not acted upon by this salivary amylase to disaccharides, that will be converted. So I told you the completion of digestion, mechanical digestion happens. The proteins, carbohydrates, and fats happens in the small intestine. So it happens like this. The remaining starch is digested. And what is the digestion of fats? What does this lipase do? Lipase converts fats to fatty acids and glycerol. To aid this action of this lipase, bile salts emulsify fats, that is, reduce the size of the globules and increase their surface area. And how this control of secretion happens? The secretion of pancreatic juice is stimulated by secretin and cholecystokinin, which is produced by the endocrine cells in the walls of the duodenum. So the presence in the duodenum of acid material from the stomach stimulates the production of these hormones. I will gist you in the end, guys, all in a continuous flow chart. You will understand what happens where. Okay. And I will talk about the liver. Liver is the largest gland in the body. It weighs about 1 to 2.3 kg. Now we'll talk about the functions of the liver. What does this liver does? Liver has various functions. It's an extremely active organ. Some of its functions which have been described, and I'll only be talking about what does the function do. Functions of the liver: carbohydrate metabolism. What is this carbohydrate metabolism? It is nothing but the conversion of glucose to glycogen in the presence of insulin, and converting the liver glycogen back to glucose in the presence of glucagon. And these changes are, which are very important regulators for the blood glucose level maintenance. What will happen? You have after a meal, okay? You have taken a meal. Then what will happen in the blood? The portal vein has high glucose content, and insulin will be converted to glycogen for storage. And glucagon converts this glycogen back to glucose whenever it is required to maintain the blood glucose level in the fasting state and all. Okay, so glucose is converted into glycogen by insulin, and glycogen is converted back to glucose by glucagon. This is the carbohydrate metabolism. That is one of the functions of the liver. Another one is fat metabolism. What happens here? There will be desaturation of fats. That is, converts the stored fat to a form which can be used by the tissues to provide the energy. Another one is protein metabolism. Nothing but the deamination of amino acids. It removes the nitrogenous portion from the amino acid that is not required for the formation of new protein. And urea is formed as the nitrogenous product, which is excreted in the urine. And what will happen when there is a breakdown of cells? Many cells die in our body, right? Every minute, every second, there will be production and apoptosis of the cells also that happens in our body. So what will happen? It breaks down the genetic material that is produced out of the broken cells of the body to form uric acid, and which is excreted in the urine. And it will also help in transamination. What is this transamination? Transamination is nothing but the removal of the nitrogen portion of the amino acids and attaches it to the other carbohydrate molecules, forming new essential amino acids. And there will be synthesis of plasmaproteins and most of the blood clotting factors from the available amino acids that occur in the liver. And what is the other function of the liver? Breakdown of erythrocytes and defense against the microbes. That is whether the liver has Kupffer cells, and they are nothing but the macrophages that will help in this function. And the other function is detoxification of drugs and noxious substances like alcohol, toxins which are produced by the microbes, and other toxins. And metabolism of ethanol. This happens only after drinking alcohol. Okay. And inactivation of hormones. Like, inactivation of insulin, glucagon, cortisol, aldosterone, thyroid, and sex hormones. Synthesis of vitamin A from carotene. Carotene is, it's a provitamin that is found in some plants like carrots, green leafy vegetables, all these things. So it will synthesize vitamin A from carotene that is available from the food that we eat. And what is the other function? The production of heat. The liver uses considerable amount of energy, that has high metabolic rate and produces heat. And liver is the main, heat producing organ of the body. Another function is secretion of bile. And this bile is synthesized from the hepatocytes, like from mixed arterial and venous blood in the sinusoids. It contains the bile salts, bile pigments, and cholesterol. Okay. And I will show you guys. Bile is secreted by the liver. Bile is secreted by the liver and is unable to enter the duodenum when the hepatopancreatic sphincter is closed. Therefore, it passes from the hepatic duct along the cystic duct to the gallbladder where it is stored. Bile has a pH of 8 and it is between 500 and 1000 ml of bile is secreted every day. What does this bile juice contain? It contains water, mineral salts, mucus, bile salts, bile pigments, and mainly bilirubin and cholesterol. What is this bilirubin? You should get a doubt. See, I'll show you what is bilirubin. It is one of the products of hemolysis of erythrocytes. This hepatic macrophages, that is nothing but the Kupffer cells in the liver, and also by the macrophages that are present in the spleen and bone marrow, there will be hemolysis. Okay. In its original form, okay, bilirubin is insoluble in water and is carried in the blood bound to albumin. So bilirubin binds to albumin and it is carried in the blood. In hepatocytes, it is conjugated with glucuronic acid and becomes water soluble before it is being excreted in the bile. Okay, unconjugated bilirubin cannot become, that is insoluble in water. The conjugated bilirubin, that is conjugation of this bilirubin with glucuronic acid, makes it water soluble so that it can be easily excreted in bile. And bacteria in the intestine change the form of bilirubin and most is excreted as stercobilinogen, okay, in the feces. A small amount is reabsorbed and excreted in urine as urobilinogen. And jaundice is yellow pigmentation of the tissues seen in the skin and conjunctiva that is caused by the excess blood bilirubin. Okay. Okay, let me see if any doubts. Okay. What are the functions? The bile salts sodium taurocolate and sodium glycocolate emulsify fats in the small intestine, excretion of bilirubin in the bile. Bile salts may make fatty acids soluble, enabling both these fat soluble vitamins to be readily absorbed, and stercobilin colors and deodorize the feces that we have seen. Biliary tract, you can see here. This is the ampulla, this is the cystic duct, hepatic duct, common bile duct, cystic duct and hepatic duct, you can see here. It is stored here. Common bile duct that will go to pancreatic duct. And this is the duodenum. This is the head of the pancreas. It is nicely fitted here. You can see this all. Bile from the liver comes here. Now we'll talk about the gallbladder. The gallbladder is a pear-shaped sac attached to the posterior surface of the liver by the connective tissue. It has a fundus or expanded end, a body or main part, and a neck which is continuous with the cystic duct. You have seen here, gallbladder. See, cystic duct is here, hepatic duct is here. What are the, and the blood supply is by the cystic artery. And there is both sympathetic and parasympathetic innervation. And what is the functions of the gallbladder? That is the reservoir for bile, and concentration of bile by up to 10 to 15 fold by absorption of water through the walls of the gallbladder. And they also release of the stored bile is also is the function of it, only stores and also releases the bile. And when the muscle wall of the gallbladder contracts, what will happen? The contracts, and then the bile will pass through the bile ducts to the duodenum, and this contraction is stimulated by the cholecystokinin, which is secreted by the duodenum, and the presence of fat and acid chyme in the duodenum will also stimulate the release of bile. Relaxation of this hepatopancreatic sphincter is caused by cholecystokinin and is a reflex response to contraction of the gallbladder. You can see here. Right hepatic duct, left hepatic duct, common hepatic duct from the liver, then cystic duct from the gallbladder, common bile duct, and the pancreatic duct from the pancreas. Here sphincter will be there, then the duodenum. Okay, all the pink color that is through liver, green color is gallbladder and pancreas. These are the ducts carrying bile from the liver and gallbladder and pancreatic juice, pancreas into the duodenum. Okay. And you can see the pancreas, the relationship between the pancreatic enzymes digest starches, that is polysaccharides, proteins, triglycerides, and nucleic acids. And what is the relationship here? You can see. You can see here. Common bile duct and pancreatic duct form the hepatopancreatic ampulla and emptying into the duodenum. Left lobe of the liver, right lobe. You can see a gallbladder, and you can see the cystic duct, common hepatic duct, common bile duct, pancreas is here, duodenum is here. So this picture gives you clearly the anatomy of all these organs. And now we will discuss about the intestinal secretions. Okay, what are these? I have told you, when the acid chyme passes into the small intestine, it is mixed with pancreatic juice, bile juice, and intestinal juice. So we have dealt about what is pancreatic juice, what will happen, and we have seen the bile juice. And now, what is this intestinal secretion? We have seen the chemical digestion of carbohydrates, proteins, and fats that occur in the small intestine. And we have seen the bile. What is the composition of bile? The bile has water, mineral salts, mucus, bile salts, bile pigments, and cholesterol. And we have seen the functions of the bile, about the bilirubin, what will happen, and all this. And we have seen the release from the gallbladder. And now we will see about the intestinal secretions. The principal constituents of intestinal secretions are water, mucus, mineral salt, and enterokinase. See, bile salts has bile pigments. Okay, and pancreatic juice has the enzymes amylase, lipase, trypsinogen, chymotrypsinogen, and procarboxypeptidase. And intestinal secretions have water, mucus, mineral salts, and enterokinase. Okay. And most of these digestive enzymes in the small intestine are contained in the enterocytes of the walls of the villi. I have shown you the structure of the villi. We can see it here. Here. This is the enterocyte. Okay. And digestion of carbohydrates, proteins, and fat is completed by direct contact between these nutrients and the microvilli within the enterocytes. And what are the enzymes that are involved in completing this chemical digestion of food in the enterocytes? They are nothing but they are peptidases like peptidase, lipase, sucrase, maltase, and lactase. What does this enterokinase do? See, guys, before telling that, the alkaline intestinal juice, okay, the pH is about 7.8 to 8. It assists in raising the pH of the intestinal contents between up to 6.5 to 7.5 also. It will go sometimes. And what is this enterokinase do? It will activate the pancreatic peptidases such as trypsin and convert polypeptides to amino acids. Okay. And the final stage of breaking down of amino acids of all peptides will occur in the enterocytes. And this lipase, what does this lipase do? It completes the digestion of emulsified fats to fatty acids and glycerol. And what is this sucrase, maltase, and lactase do? They complete the digestion of carbohydrates by converting disaccharides such as sucrose, maltose, and lactose to monosaccharides. So initially in this digestion, what will happen? There will be mastication, deglutination, all this happen. The mechanical digestion happen in mouth, pharynx. We have seen all that. After that, when the food passes to stomach, there will be action of HCl, and there will be no complete mechanical digestion does not happen. There will be formation of chyme that will be coming to duodenum. There will be secretions of pancreatic juice will be there, bile juice will be there, intestinal juice will be there, where it gets mixed when it's passing through the duodenum. And with all these things, the remaining digestion of carbohydrates, proteins, and fats will occur in the intestine, which is remaining. The remaining digestion, some would have been left out. The complete digestion is not happened, but the mechanical digestion will be completed in the intestine by these enzymes, these peptidases, lipases, like, and sucrose, maltase, lactose. Okay, so peptidases will complete the protein digestion, lipases will complete the fatty acid digestion, that is the emulsified fats that which are remaining, and sucrase, maltase, and lactase will complete the digestion of the remaining carbohydrates. How these secretions are controlled? This mechanical stimulation of this intestinal glands by the chyme is believed to be the main stimulus for the secretion of intestinal juice. Although the hormone secretin also may be involved. Okay. Now we'll discuss about the absorption of nutrients. So intestine will have absorption of nutrients. It occurs by two possible processes. That is nothing but the diffusion and active transport. So you can see the villi here. The lumen of intestine is full of villi. See, here there are carbohydrates. These circulated ones are polypeptides. These are fat molecules. This is the goblet cell, the blood capillaries. You can see all this structure very clearly. So what will happen? Diffusion. This monosaccharides, amino acids, fatty acids, and glycerol diffuse slowly down their concentration gradients into the enterocytes from the intestinal lumen. And active transport. What is this active transport? These monosaccharides, amino acids, fatty acids, and glycerol, they may be actively transported into the villi, and this is faster than diffusion. Disaccharides, dipeptides, and tripeptides are also actively transported into this enterocytes where their digestion is completed before transfer into the capillaries of the villi. And monosaccharides and amino acids, they'll pass into the capillaries in the villi, and fatty acids and glycerol into the lacteals. See, this is the lacteals. Fatty acids will pass here. And monosaccharides and amino acids into the capillaries in the villi. You can see carbohydrates, capillaries. Okay, you can see the picture clearly. These are the carbohydrates. These are the polypeptide molecules. You can see the blood capillaries here, and lacteals are here. And some proteins are absorbed unchanged. That is like antibodies that are present in breast milk and oral vaccines. That some vaccine like poliomyelitis vaccine. All this. The extent of protein absorption is believed to be limited. And other nutrients such as vitamins, mineral salts, and water are also absorbed from the small intestine into the blood capillaries. And fat soluble vitamins. What are these fat soluble vitamins? Vitamin A, vitamin D, vitamin E, and vitamin K. They're absorbed by the lacteals along with fatty acids and glycerol. And I told you vitamin B12, it combines with the intrinsic factor in the stomach and is actively absorbed in the terminal ileum. Okay. And the surface area through which absorption takes place in the small intestine, it is increased by this circular folds, okay, of mucous membrane and by the very large number of villi and microvilli present. So it will provide a greater surface area for the absorption of food. And when they calculated, they found that the surface area of small intestine is about 5 times that of the whole body. A large amount of fluid will enter the alimentary tract each day. Of this, only about 500 ml is not absorbed by the small intestine, and then it is passed through the large intestine. Any doubts, guys? Can you hear me? Which part was not audible?
No, we can hear you, Dr. Pravallika. That is long back.
Was it all clear?
I think so, yes.
Guys, you should understand this to understand the next part. And homeostasis is very small, but large intestine will take some time. I'll explain you large intestine and remaining part in next class. That will just take another 10-15 minutes. But till here, it should be very clear that people should be clear about this pancreatic juice, bile juice, the constituents of all these things. You just type me with your doubts. I'll stop sharing and I will share you with another slide to quickly revise you. Dr. Ajit, you can ask them to share their doubts if any doubts are there.
Yes, guys, you can please write your doubts and you can speak one by one now. Please put your doubts if anyone have that. You can speak. Dr. Pravallika, we have something in chat box.
Ma'am, we can't hear you. Hello, ma'am.
Now can you hear me?
Yes, ma'am.
We have learned about the gastric juice, right? Initially, in that picture. See, pepsin and gastric lipase, both of them are secreted by the chief cells in the stomach, which converts proteins to peptides and here triglycerides to fatty acids and monoglycerides. And pancreatic juice today we have learned about. Here it converts starches to maltose, maltodextrose, and dextrins. Please mute your mics, guys. And proteins to peptides and chymotrypsin also converts to peptides. We have seen these enzymes, carboxypeptidase, which will convert amino acids to peptides, and pancreatic lipase, the triglycerides, and ribonuclease, deoxyribonuclease, and it will form the formation of nucleotides from RNA, that is ribonucleic acid and deoxyribonucleic acid. One second, some question is there. Okay, it's not from the lecture, that's okay. Somebody asked me, yes. Where does most of the digestive process takes place? See, mechanical digestion will take place in the, so digestive process is different, right? Mechanical digestion, chemical digestion, all this. And if you have seen, after the food is properly masticated only, it land in the stomach, there will be action of hydrochloric acid and all this gastrin will be there, it will stimulate, and all the contents will be converted to chyme. There the complete digestion will not take place. But in small intestine, whatever is remaining, the complete chemical digestion happens in small intestine. So we can take it as most of it happens in the small intestine. Okay, actually one of my colleague child who is 3 years old is suffering with 6 mm solitary gallstones. It is growing day by day. Seema, you are telling that there is a gallstone and all. See, all this will be decided by the Department of Surgery. Only surgeon knows what is the condition of the 3 years old kid and how big it is. You know, it will be a surgical option at what it should be removed or operated, and you are telling like this, and it should be gone according with the opinion from the surgery. It goes on its own. That all I cannot tell, because it all depends what is the content of the stone, size of the stone, and it should be gone through surgery. Even medical department people will not take any decision. Surgery part, surgery people only will take care. So vaguely, we should not suggest anything to any kid, and it is related to pediatric surgery. So a pediatrician along with the pediatric surgeon will take the decision. And causes of hepatomegaly, you will have so many reasons. It may be, hepatomegaly may be because of infections also. It may happen. So many reasons will be there. Even the accumulation of triglycerides. There also there will be increase in size of the hepatic cells because of production, the accumulation of triglycerides. As it progresses towards cirrhosis, also you will see hepatomegaly in even some infections, hepatitis. You'll see elevation of liver enzymes, and cancer also you will see so many multiple conditions causing hepatomegaly. Any other? We will see the negative feedback mechanism after we finish the large intestine. You'll understand everything. I will surely discuss with you these things in the next class. Okay, guys, for today we'll end up the session. If you have any doubts, please post it in the group. Suppose you may not get doubts now, but when you go through the class and read through the textbook or anything in while you are reading or listening to the class, at that time you will get doubts. You can post it in the group. We'll discuss it in the next class, or I will answer you back in the same when you ask me in the chat box, or you can do it in the WhatsApp also. You will surely get doubts. Please post them in the group. I'll clear all your doubts, because when you know the digestive process and all these things, so when you're reading about the disorders of the gastrointestinal system or something, then you can understand why there is nausea, what will happen when there is, you'll understand the diseases better. When there is when there is some damage to pancreas, there will be no release of pancreatic juices properly. So which enzymes are not released? So if that enzyme is not released, what is going to happen? So to understand the diseases, okay, you should understand the actual function of these enzymes properly. Don't go in too much deep. See what is the constituents of pancreatic juice, what is the composition of bile juice, what is the composition of intestinal juice, what is happening in small intestine, what digestion is happening. So which enzyme is responsible for the digestion of what. So what will happen when you know which enzyme is responsible for what? You will understand what will happen when there is deficiency of that enzyme in the disease, and what is the role of doing that test in case of the organ dysfunction. So you are seeing amylase, you have to go for amylase. They'll tell, doctor will tell, go for serum amylase test, go for this test, go for this test. So why are this doctor suggesting to go for a specific test? So what organ abnormality we have? Yes, histamine do affect the mucus secretion and also gastrointestinal mucosa. Here H2 receptors. Okay. I've shown you in this picture. I'll show you again if you want.
So if you have any questions, guys, you can even post in the groups. We will take it
Yeah,
later.
I will post the answers in the WhatsApp group. I'll send you the answers that will be more clear and specific, and other people also can read if they missed out while reading. But please concentrate on all the topics what I have told you, and what is the role of each enzymes and how it will be helpful for you to understand the diseases.
Absolutely.
You have 1 week, but you have 1 week time The simple, don't go into too much complication. Read what is the function, what is the enzymes, which organ secretes, what is the composition, what is the role. Don't confuse with all the things.
Right.
Okay. I'll post if anything is there not answered in this class, please post it there. I'll post you the answers.
Yeah.
Okay,
Okay.
guys. Thank you.
Thank
See
you
you.
guys.
Have a good day.
Take care. Have a good day. Bye guys. Take care.
Hmm.